Hello everyone. A very happy evening to all the students here. So today I welcome you all to the day 2 of refresher course. So today we'll be discussing a topic of biology, which is transcription and genetic code. And yes, Umesh, good evening. So today we have with us Dr. Ajith Madhavan, who is an associate professor from Amrita School of Biotechnology. So I welcome you, sir, towards today's session. And
Good
to
evening,
all
ma'am.
this,
Thank you.
yeah. So to all the students who have been waiting, I would just give a brief idea about who sir and where he's working. So he's actually from Amrita. He's working under the School of Biotechnology, which is in our Kerala campus. So he has completed his master's in applied microbiology and he holds a PhD from the Bharatiyar University in the area of enzymology. So he guides a lot of students, I would say more than 200 students in their projects and all the other activities. So thank you so much, sir, for coming. And dear students, please note that the session is exclusively for the biology students. And if you're having any queries, please do type in the chat box so that we'll be addressing all of them towards the end of the session. So thank you so much, sir, for coming. And yes, I think we can just start.
Yeah, ma'am. We can start, ma'am. Okay. Thank you so much, ma'am. Thanks for the participants. In fact, it's an honor to interact with them. Shall I share my slides, ma'am?
Yes, yes.
Very good evening. In fact, I thought of having a small discussion with you people. If time permits, let's go deep into it. But the objective of today's session is to sort of get inspired by the molecular mechanism that happens inside the cell. So in fact, I can't even stop getting interested, in fact, getting intrigued by the complexity of the molecular mechanism present inside the cell. And moreover, in fact, those mechanisms which apparently seems to be designed, seems to be performing a function as if somebody has designed it, but all happens very randomly by the process of thermal motion and so on. So I would say sort of an order developed from chaos. Inside the cell, a small 2 micrometer cell, 10 micrometer cell, we have the cytoplasm, different components dissolved in water, and randomly they interact and bring about amazing functions. So today we are going to talk a little bit about those functions. So in fact, let me start from here. So we know that the information that is required to build the cell, the information that is required to functionalize the cell, each cell has its own functions. You have around 30 trillion cells in your body, and they can be grouped into cells of different types, be it the nephrons, be it the neurons, be it the epithelial cells, be it the cells that goes into the making of your heart, and so on. So they have different functions. Those functions are due to the information that is coded in the nucleic acid, especially the double-stranded DNA molecule. It's really interesting. If you look at the DNA molecule, if you look at the width of the DNA molecule, it is a material. In fact, it has dimensions associated to it. It has length, it has width, and so on. So if you look at the width of the DNA molecule, it is uniformly 2 nanometer width. And if you look at the length of the molecule, in the eukaryotic cells, if all the chromosomes are joined end to end, if you tie them end to end, it's around 2 meters long. In the case of a simple bacteria, it is around 2 millimeters long. But the problem is this large string of DNA has to be packed inside a small space, 2 micrometer in the case of bacteria, for example, or 10 times, 20 times the size in the case of eukaryotes. So it needs to be packed. So you know the problem associated with the phone cord, is it not? So nowadays it is all wireless, but you would have surely seen the handset of a phone connected by a cord. After multiple usage, you can see the cord, the wire of the cord, I mean, the cord gets entangled. It's very difficult to entangle them. It gets tangled. That's the idea. But in the case of genome, the total DNA content, you call it as a genome, that is packed inside a small space, goes through many molecular mechanisms. One of them, of course, is transcription. Then it has to be replicated. It has to be copied. And it has to be expressed, starting from transcription to translation. It has to be expressed. So it shouldn't be tangled. It shouldn't be knotted. It should be accessible. So it has to be packed neatly inside the cell. So that's where the condensation of the DNA molecule plays an important role. So I'll give you some basic aspect of it. These are the things probably you would have got introduced to, but it's really interesting to see how this is packed inside the cell. They are neatly packed and orderly packed and placed inside a particular functional area called as nucleus in the case of eukaryotes. In the case of prokaryotes, though you don't have a nucleus, there is a small area called as nucleoplasm, where it is beautifully arranged in a patterned form and attached to the membrane and placed there. So I'll give you some interesting information about this packing. So you have a string of DNA, it has to be condensed so that it fits into a smaller space. So some of the basic details about this packaging is, if you look at, on an average, around 5 centimeters of DNA is condensed into 60 to 70 millimeter chromosome. That's the condensation that we are looking at. So 5 centimeters of the DNA is condensed, packed orderly in an orderly fashion. Certain regions are tightly packed. You call it as heterochromatin. Certain regions are loosely packed. Euchromatin, U meaning true, euchromatin. Most of the genes seems to be concentrated in the euchromatin, not in the heterochromatin. Probably something to do with accessibility of those regions. So 5 centimeters of the DNA is packed in 60 to 70 millimeter of chromosome. So what are you looking at? So before going into what we are actually looking at, in fact, how it is condensed is the interesting question that we can ask. So if you look into a sewing machine, you can see there is something called as a bobbin, a spool on which a long thread is wound. Likewise, similarly, in the case of DNA molecule also, you have a spool on which the DNA is wound. So the spool along with the wound DNA, you call it as a nucleosome particle. You call it as a nucleosome particle. So it is a spool on which the DNA is wound, and that particle, the particle is made up of protein, and over the protein, the DNA molecule is wound. So a larger stretch of DNA is wound on spools. It's spooled. So these particles are called as nucleosome particles. So how much of DNA is wound? That's an interesting question that you can ask. Around 140 to 150 base pairs of DNA are associated with these particles. So are there space between these spools? So you have a spool, a circular structure on which the DNA is wound. You have another. Is there a space between them? Is there a distance between them? Yes, around 50 to 70 base pairs. They are called as the linker DNA. So in this nucleosome, so you along with the linker molecule that separates the two nucleosome, around 190 to 120 base pairs are condensed in this particular fashion. In fact, I'm not going into the diagram on your left-hand side. It actually is the experiment to understand how much of DNA is condensed, how much of DNA is wound around the histone. It's a very simple experiment. It is called as a nuclease protection experiment. Any DNA that is associated with the spool, which is made up of protein, it is protected from digestion. So what you can do is digest it. You get a spool with wound DNA in it. Remove the protein by enzymes protease, for example. Then remove the DNA and find out its length. That's how people arrived at 140 to 150 associated with the spool, and 50 to 70 that connects the spools together, linker molecule. So what are these spools called? They are called as histones. They are called as histones. They are octameric proteins. Octameric meaning, octa meaning 8. It is made up of 8 proteins. So just a spool, it forms a circularly oriented a protein molecule, a protein complex on which the DNA is wound. So this is only the first level of condensation. Naked DNA is wound around the histones. Histone is an interesting molecule you people should study whenever you get time. You people should understand it. Histone modification is responsible for functionalizing the DNA. Epigenetic modification, people call it. It is not genetic, it is epigenetic. So those are interesting things that you can study about histones.
Sir, actually, sorry to interrupt. There is a question which is from Khushi. Can you explain what exactly is non-histone protein?
Non-histone, I didn't get you.
Non-histone? Yes. I'll just a second.
Okay.
So I think I have given you permission to talk, Khushi. You can just unmute and talk directly with sir. I saw a query from you.
Khushi, I understood your question, and if not, you are not able to speak, fine.
Yes.
In fact, the histone proteins are those proteins which goes into the making of the circular complex, the spool-like complex. Tell me.
Good evening.
Yeah.
My question is that what is the function of non-histone protein?
Okay, so in fact, non-histone protein, I talked about only histone proteins. There are some proteins which are not part of this complex per se. They are not part of this complex per se, but they are associated with this nucleosome. So you can see that red protein that you can see, which is called as the linker proteins. The linker proteins is not directly associated with the histone. Rather, it is only it helps to stabilize the nucleosome. So those proteins are called as non-histone proteins.
I hope your query is cleared. I think so. So Manasi, I don't know whether you have any queries for sir. I have just given you permission to talk because I have seen you raising your hands. So if you have any queries, can you please unmute and discuss it with sir? Manasi, I hope I'm audible. Yes, sir. I think we can continue. Okay. She has just replied in the chat box. We can continue.
Okay. So as I mentioned, naked DNA gets spooled onto the histone. Then the first level of condensation. And after that, the fibers that they form, they get condensed again twice at two levels. One, they get condensed, for example, into a helical structure. Then the helical structure again winds over itself to condense into a chromosome. So there are three levels of condensation. The first level is important because it winds around the histone. Then it forms helical structures, solenoid structures, and so on. And after that, it winds up into a cord-like structure that goes into the making of the chromosome. So the individual chromosomes, of course, they vary in the level of condensation. Their sizes also varies because the length of DNA that goes into the making of the chromosome also varies. So this pattern of condensation, as I mentioned before, produces the heterochromatins and the euchromatins. In fact, the heterochromatins and euchromatins of a particular chromosome is also a constant feature. It is one among the features that helps to stain the chromosome differently. There are different ways of staining a chromosome because the staining gives you an understanding about the composition of the chromosome at the chromosome level. The fine structure of the chromosome can be understood. In certain instances, part of the chromosome will be lost in certain genetic diseases, or certain chromosome gets fused together. So you call that as chromosomal aberrations. So chromosomal aberrations can be understood by staining the chromosome. So this heterochromatin and euchromatin, they stain differently. So those are the applications, those are the features of the chromosome, which in fact can be used for understanding the aberrations, understanding the anomalies associated with chromosomes. Now, having done that, we'll now go into another interesting domain altogether. So we know that the information is a two-dimensional information in the case of a DNA molecule. It is a sequence information. Like any other languages, the DNA is also written in a letter of 4, in our case. Very simple language it is. Adenine, guanine, thymine, and cytosine. Adenine and guanine, you all know that falls into a group called as purine, and thymine and cytosine into pyrimidine. So it is also interesting to see that why a purine combines, or what are the implications of a purine combining with a pyrimidine, and vice versa. Because you know that the purine is made up of 2 aromatic rings. At the same time, pyrimidine is made up of 1. So you have a bulky and a less bulky nucleotides. When they combine together, they form a particular width. So in any combination for that matter, the width of the DNA doesn't change. Imagine a scenario, if pyrimidine and pyrimidine are combining, the width will be smaller. If it was purine and purine combining, the width will be larger. The backbone of the DNA would have been differentially, would have had differential widths. The width is very, very important for the function of the DNA. That's how there are different proteins that associate with the DNA, for example, to find out, are there any mismatches? Is there any anomalies associated with the DNA molecule? The backbone gets skewed. How do you know that the backbone gets skewed? Because the width of the DNA is uniform, 2 nanometer. So that also answers that question, why the width of the DNA is always uniform. It is because the combination of a lesser bulkier and a bulkier bases together. So that's a digression. But the main important point is, even before unraveling, the replication mechanism, the information transfer mechanism in the case of biological system exhaustively Francis Crick, the co-discoverer of the double helical structure of the DNA, he went on to say that the flow of information in molecular biology always is from DNA to that of RNA to that of protein. So the flow of information in molecular biology is actually defined by a central dogma. The flow of information is always from the DNA to that of RNA to that of protein. So it was only proved later. This dogma seems to be unchallenged to a larger extent. And it also seems to be flowing in one direction, DNA to RNA to that of proteins. But nobody has seen a complete reversal of information from protein to that of RNA to that of DNA, but partial flow of information from RNA to that of DNA, people have seen. So if you consider the flow of information from DNA, which is a nucleic acid, to another, a sister nucleic acid, I would say, RNA, is transcription. The flow of information in the reverse is called as reverse transcription. Certain viruses, they do that. Retroviruses, you call them, including your HIV and so on. Retroviruses do that. The flow of information is from, at least part of the life cycle as a flow of information from RNA to that of DNA. But that's not the point. It flows from DNA. The information flows from DNA to that of RNA to that of proteins. So the flow of information from DNA to RNA is called as transcription. And the flow of information from RNA to protein, it is called as translation. Transcription meaning, without changing the phonetic of a language, you just write them in a different language. So you can, in fact, whatever that I speak now in English can be written without changing the phonetics in any other language. For example, in my mother tongue, Malayalam, or in Hindi. So this process is called as transcription. One information is slightly rewritten into another information. But if an information is interpreted in a different language altogether, you call that as translation. So the information written in RNA, if it is converted to an information written in amino acids, you call them as translation. It's rewritten. So that is an interesting question that we can ask. DNA is nucleic acid. RNA again is nucleic acid, but a slightly variant of the raw material that goes into the making of the DNA. Here you call it as ribonucleotide. There you call it as deoxyribonucleotide. Because a 2' OH group is missing in the case of DNTPs, or deoxynucleotide triphosphates. So the sugar is little bulkier here. Moreover, instead of thymine, you have a slightly varied form called as uracil. So if adenine base pairs with thymine, in the case of RNA, adenine base pairs with uracil. So that's the change that you see. Otherwise, it's all nucleic acid. In fact, the DNA doesn't recognize whether it is adding RNA or ribonucleotides or deoxyribonucleotides, which is, for example, in replication, hybrids are formed because the primer that is attached is an RNA molecule. So the machinery doesn't know, but there are disadvantages associated in having a hybrid, so that the cell takes elaborate measure in order to remove them. You would have seen that. Because otherwise the hybrid DNA becomes unstable. It is because of the 2' OH group. It makes it more hydrophilic. So hydrolytic action is more on the RNA molecule than on the DNA. So it is more unstable than the stable DNA. So the question that I'm asking is, why do you require an RNA intermediate? If both of them are nucleic acid, the proteins, the information can directly be translated. Why there is a stepwise expression in information from DNA to that of RNA to that of protein? One of the main reasons is regulation. So the information needs to be regulated. So the best example that you can see in the humans, for example, in different cells are, I told you there are 30 trillion cells and different types of cells. And all of those cells have the same complement of DNA. But if you compare a neuron and a nephron, they are structurally and functionally different. So the information is the same. The retrieval of the information is different. Spatio-temporally, the information is retrieved at a particular time in a particular of a particular gene. So some of the genes are on, some of the genes are off. You have nephron. A different sets of genes are on and a different sets of genes are off. You have a neuron. So transcription is very, very important. It regulates the flow of information. Whether a gene should be switched on or off, at what level, it depends on the transcription process. That's why the transcription is so important. So I'm going into the transcription process.
Sir, actually, yeah, just a second, sir. There is one more question. What is the reason that only retrovirus are able to produce DNA from RNA and other organisms containing RNA are not able to do so? I think I'll just give them permission also. It's
okay, that's an interesting question.
Okay, I have given you permission. I think it's Pugal. I need some Pugal. Okay, I have given you permission. Can you please directly communicate with sir?
Yeah.
Please try to talk. I have. You can unmute and talk now. Okay, sir. Yes.
Pugal.
Okay, I think.
You want to ask the question to that?
Yes, yes. That's the question that I'll just read once again. What is the reason that only retrovirus are able to produce DNA from RNA and other organisms containing RNA are not able to do so?
That's an interesting question. In fact, I would say I could only answer. I could only guess what could be the reason for that. For example, the viruses, especially in the case of retroviruses, or in case of any viruses for that matter, certain viruses adopting the reverse transcriptase, not the other. There are some associated sort of advantages to it. Of course, there is, but still you can that advantages doesn't explain fully why some has reverse transcriptase and the other doesn't. So, for example, you have the DNA viruses, and the DNA viruses, they produce the RNA molecule, and the RNA molecule can be maintained in high copy number. Instead of copying the whole genome, the RNA molecules can be maintained in high number. It has to be maintained in high number. Why? Because transcription has to happen, then translation has to happen, and it has to produce the protein. It's already there in high number. So the copies are ensured. You have high numbers of RNA molecule. What you next have to do is, you convert the RNA molecule to the DNA molecule. So certain type of RNA molecule goes into the making of the protein, certain goes into the making of the DNA molecule, which is its genome. So most of the viruses, in order to increase the number of the progenies, they have to improve upon the strategy to make more copies of the nucleic acid. So I personally feel that the reverse transcriptase utilizes the high copy number of the RNA molecule that is made so that it can churn the DNA molecule in high number. So that only answers the partial aspect of your question. But in certain instances, I can give you an interesting example, which is more disadvantageous for the organism than advantageous. For example, there are RNA viruses with plus strand and minus strand. So the plus strand is the sequence that shows similarity to the mRNA molecule. It is similar. The negative strand is complementary. Now you understand what is the problem. In the plus strand viruses, it can directly act as an mRNA molecule. In the negative strand, it has to be converted to positive, then it has to produce the protein. So don't you see there is a disadvantage for the negative sense RNA molecule? So if you ask me a question, why did that virus did not resort to the same strategy? Because it is the evolutionary journey that it has taken. So that could be the only answer that I can give you. But the first answer partially answers your question. To increase the number of the genome so that the number of progenies can be increased.
Sir, actually, there are two comments also. One is like retrovirus got reverse transcriptase. Only retrovirus got reverse transcript. And another is a question. If the gene has to be regulated, why is the intermediate only in RNA form but not in DNA form?
That's an interesting question. I'll ask the first question. I'll answer the first question first. In fact, no. Retroviruses is one group of animal viruses that has this reverse transcriptase activity. But even the viruses that infects bacteria has the reverse transcriptase activity. There are RNA reverse transcriptase activity viruses, DNA reverse transcriptase activity viruses, RNA RT viruses, they call it. DNA RT viruses, they call it. So that is the answer for the first question. The second question, that's a very interesting question. In fact, see here, in a DNA molecule, the genes are physically attached. The genes are physically attached. So you know that the problems associated with it. Can you physically sort of contextually remove the genes out and express them and selectively express the proteins? Because the copy number is not there. But in the case of RNA transcription, you are basically amplifying that gene. Though there is only one gene, by transcription, you produce umpteen number of genes with different retention times, or people call it as half-life. So if the half-life is higher, more proteins will be produced. If the half-life is lower, lesser amount of proteins will be produced. If the genes are not transcribed, the gene is switched off. If the genes are transcribed in higher quantity, it is switched on or expressed. So having an intermediate has more advantages than working on the DNA molecule itself. Hope that answers the question.
Okay, it's like there is an addition. Why is it in RNA form? To the same question, I think that you've replied. There is a.
Okay, that's a good question. Why it is in RNA form? That's an interesting question too. For example, in fact, those are really interesting questions. I can only make guesses from my educated guesses, I would say. For example, RNA molecule has its advantages because why RNA in transcription? Why not DNA? Because RNA is less stable than DNA molecule. Because the stability of the RNA molecule is important, because it is fleetingly present in the cytoplasm. For a few minutes it will be present, it is gone. So it serves that particular purpose. So it is not very stable because stability is not required. As I told you, the 2' OH group makes it more hydrophilic, so that it is more prone to hydrolysis than the DNA molecule. That's why previously even people talked about an RNA world. People even believe that some of them very strongly, but there are evidences to that. The RNA molecule was the first molecule that was used to store information. But the RNA molecule showed a small variation by removing its 2' OH group to form the DNTPs, which combine together to form a slight variant of the RNA molecule. So it had more stability. So the DNA molecule was graduated as the main information storage, and the RNA molecule for storing the information in a fleeting fashion for regulation and so on. Even nowadays, you can see such molecules being present in the primers, for example. In replication, you may ask the question, why RNA is used as a primer? So people say that it is the sort of a vestige that is present, which indicates the RNA world altogether. The ribosomes elaborately has the RNA molecule. You call them as snRNA or SNRPs. It has structural function. It also has informational function. So you can see the RNA function of the RNA molecule has been religated for something because it had a lesser stability than the DNA molecule. So the answer is, in a nutshell, the answer is the instability of the RNA molecule and its ability to make more copies due to the available machinery. It is more suited for regulation than storage of information.
So yes,
Okay,
I
is
think
that clear?
that is clear. Yes, because he has typed that in the chat box. So I think we can continue.
Okay.
Yeah. Thank you.
So transcription, as I told you, the information is rewritten in a different molecule. But it is also interesting to see that there are 2 strands associated with the DNA molecule. There are 2 strands. It's a double-stranded DNA helix. But only one of the strands has the information to be converted to be transcribed into an RNA molecule. So the other strand is not transcribed into an RNA molecule. So this is beautiful. Why? Because only one strand has the information to code for the RNA molecule. But in the case of replication, it is different. Both the molecules, they separate. Both of them, they act as a template on which new strand is synthesized. So the 2 strands become 4 strands and so on. So in this case, only one single strand is synthesized from one single strand of DNA molecule. So how does the system know which strand to transcribe? That's an interesting question. I'll answer that little later. So this gives these 2 pictures, gives you an idea about the transcription. Also, it gives you an idea about something else as well. So this is a double-stranded DNA molecule. The double-stranded DNA molecule, they open up, and one of the strands act as a template. And on the template, if the DNA has adenine to it, a uracil will be added. If the thymine, adenine. If it is guanine, cytosine. If it is cytosine, guanine, and so on. You know the Chargaff's parity rules, which says that a purine base pairs with pyrimidine. If it's adenine, thymine, or uracil. If it is guanine, it is cytosine, or vice versa. So I told you this information gives you another additional information as well. So if you ask the direction of synthesis of the RNA molecule, it is again in the 5' to 3' direction. It is again in the 5' to 3' direction. as in the case of the DNA has direction, is it not? The strands of the DNA, the polynucleotide chains of the DNA has direction. One has a phosphate at its end, the other has OH group at its end. So you have a 5' phosphate end and the 3' OH group. So anybody can ask a question, what is the direction in which the RNA molecule is synthesized? 5' to 3' direction. So the 3' is actually growing. One nucleotide at a time, it is attached and it is growing. So see here, central to the synthesis of the RNA molecule from a DNA molecule is a polymerase. As the name indicates, polymer meaning a collection of monomers is called as a polymer. So this polymer of RNA molecule is made by an enzyme called as RNA polymerase. So the RNA polymerase is a shortened word of otherwise it is called as DNA dependent RNA polymerase. So that gives you an idea about distinguishing the RNA polymerase from the transcription from the reverse transcription. In the reverse transcription, you have to have an enzyme called as reverse transcriptase, which is called as RNA-dependent DNA polymerase. So you can see the difference. So technically, it helps you to differentiate different types of polymerases. So the polymerase involved in transcription, it is called as DNA-dependent RNA polymerase. It depends on the DNA template and synthesizes an RNA molecule. Hence, it is called as DNA-dependent RNA polymerase. It has a crab claw-shaped structure. So it has a crab claw-shaped structure. So from the ribbon model of the structure, this is, you can see roughly a crab claw-shaped structure. It has pincers, 2 pincers. Then for the transcription and the RNA polymerase, there are slight variations with regard to the prokaryotes and the eukaryotes. The prokaryotes, that is constituted by eubacteria and archaea. So these RNA polymerases are made up of different protein subunits. They form complex. So these are the complexes that you see. For example, in the case of bacteria, you have the beta subunit. Then in the case of the, in the case of bacterial RNA polymerases, you have a beta prime, the beta subunit, and so the alpha subunit, and so on, the omega, and so on. So likewise, in the archaea, it is different. In the case of eukaryotes, you have RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and so on. RNAP1, RNAP2, RNAP3. They have different functions, encoding mRNA, encoding rRNA, encoding tRNA, and so on. So they are made up of different subunits, RPA1, RPA2, and so on. So if in the case of prokaryote, it is plus 6 proteins. In the case of RNA1, it is plus 9 proteins. In the case of 2, it is 7 proteins. In the case of 3, it is 11 proteins, and so on. So it's just made up of complex. The idea is very simple. It has a crab claw-shaped structure. It is made up of subunits. So see here, the transcription, as you all know, in fact, for better understanding, for better grasping the concept, you can basically divide them into 3 phases. as such is a continuum, but you can divide them into 3 phases so that you can understand the concept better. You can understand the molecular mechanism better. The first one is called as initiation. The second one is called as elongation. And the third one is called as termination. Only 3 steps. You have initiation, elongation, and termination. So each step is very, very important. Why? Because each step leads to the next. If the first step doesn't happen, the second and the third doesn't happen. If the second is not initiated, the third doesn't happen, and so on. It is a cascade. The first step, the second step, and the third step. Amongst these 3 steps, the first step is very, very important, because I told you what differentiates a nerve cells from a new nephron. It is the differential switching on and switching off of genes. So it has to be timed properly. If it is not timed properly, then the development doesn't happen properly. The structure and the function will be lost. So which gene to identify, which genes to transcribe, which not to transcribe, has to be decided in the very beginning itself, in the initiation itself. So how does the system know which genes to transcribe is the question. Or how does the system know which is the starting point of the gene which has to be transcribed? So initiation is a very important step. And of course, equally important are elongation and termination. So let me revisit. I'll talk about these steps in detail, then come back, revisit, and let's walk through these 3 processes a little later. So listen very carefully. So the question is the question is very simple and straightforward. How does the system know where to start? And of course, when to start? Which gene should be initiated in transcription? Which gene has to be transcribed? That's the question. So see here, the gene has a set of start signals that helps the that helps the transcription machinery to identify the start point. It also helps to identify the end point, the start point and the end point. So the initiation or the first phase of transcription happens in the start point. It has to know where to start, because you know that if one nucleotide is not read properly, then the whole information is gone. So every nucleotide in its same order has to be read. So it should know where to begin. So see here, I'm looking at a gene. I'm looking at the start of the gene. I'm looking at the start of the gene. So that particular start point has certain features that helps the transcription machinery to identify it. These features seems to be very common across different organisms, across different genes. So there seems to be some consensus in the structure. Consensus, the gene seems to agree to these structural features. So these are called as consensus sequences. They have some consensus pattern. So very quickly, let me tell you what are those consensus. What is the pattern that you see in the start point? How does the transcription machinery know where to start? So for example, you are riding on a long road, and the milestone serves as certain sort of information for you. This is the start point, and this is where you have to reach, and this is where how far I have to go, and so on. So these are sort of signposts which helps the machinery to decide where to start, how long to go, where to stop. So listen very carefully. So the start point is called as a promoter for the minimum signal that is required for the transcription to start is called as a promoter. Of course, there are many other sequences that are required. Enhancers, repressors, insulators, operators, and so on. There are many other sequences. I'm going to only talk about that bare minimum sequence that is required for the transcription machinery to identify. I'm looking at that point. Where does it start? So see here, this is how it looks. The pink colored region, this is the DNA molecule, the double-stranded DNA molecule. The pink colored region is called as the -35 region and -10 region. You may ask me, what is -35 and -10? So in a number line, you know, something towards your left, you name it as left to the 0, name it as number with a minus sign associated to it. Towards your right, you name it as plus. So with respect to what, it is called as -10 and -35. With respect to the point, the nucleotide which is transcribed first, the first nucleotide that is transcribed. So all the sequences that is waiting to be transcribed towards the right is called as the downstream region. And everything that won't be transcribed, but which helps to initiate the transcription, is called as the upstream region. So the reference point is the nucleotide, the first nucleotide that will be transcribed. Towards the right, you have more nucleotide that needs to be transcribed. Towards the left, you have the signal. So from +1, you have the -1, -2, -3, -4, -5, -6. The -10 region is a very important region. The region around that -10 nucleotide is very important. The sequence around the -35 is very important. Then there is another sequence indicated in green, which is called as the upstream element. I told you upstream. So you imagine like a stream flowing from the mountain to the valley. The stream in the mountain, you call it as upstream. When the stream flows into the valley, you call it as downstream. So likewise, you have an upstream region and a downstream region. So what is the sequence that is present in the -10 and -35? You need not memorize it. Just understand it is called as a consensus sequence. Consensus meaning most agreed upon sequence. Most of the promoters have that sequence. What is that sequence? TATAAT. People call it as Tata box. Since it is a box, you call it as Tata box. Then in the -35 region, you have TTGACA and so on. In the -35 region. Then very importantly, listen very carefully, I will not be able to have time to explain to you. The distance between these two regions are important. It has to be around 17 to 19 nucleotides. So if a transcription has to start somewhere, you have to have the sequence. A -10 region, a -35 region, they have to be separated by 17 to 19 nucleotides. And in certain instances, upstream elements should be required. There are some variations to this. I'm giving you the structure of a typical promoter. There are always variation in biology. So this is the sequence that is required for transcription to start. So how does it start? Somebody has to recognize this promoter. So promoter is called a promoter because it promotes transcription. So somebody has to recognize it. So who will recognize that? The polymerase will recognize that. The polymerase is the one that transcribes the DNA molecule. It's just the one should recognize the start point. It recognizes it. So you people are mature enough to understand that it is not the polymerase that is recognizing it. It has a component present in it, which is called as a sigma factor. The sigma factor is a subunit. It's a protein that is associated with the polymerase. So the polymerase along with the protein is called as a complete enzyme. In complete enzyme, in biology, it means holo enzyme. It is called as holo. Holo meaning complete. The enzyme will be complete only if it is a sigma factor is present. Now you know what is the function of the sigma factor. The sigma factor is the one that goes and identifies a particular promoter in a particular time. So you would have correctly guessed by now that there are different types of sigma factors. The sigma factor, for example, they are called as sigma 70, sigma 24, sigma 38, and so on. There are different sigma factors. These sigma factors specifically identify a certain group of genes. Now I have answered partly, how does a particular gene get transcribed in a particular cell in a particular moment in time? Apart from promoter, you have to have different sigma factors along with different other sequences to identify at a particular moment in time which gene to transcribe and which not to. That makes a huge difference into a cell being a nephron and another cell being a neuron. So this is called identification, the initial step. Now I'll add another level of information. Listen very carefully. If you take an RNA polymerase, I told you roughly it looks like the claw of a crab. So I told you pincers, there are two pincers, beta prime pincer and beta pincer. Now this diagram, you need not go into detail. This is little more technical, but what you need to understand is the RNA polymerase has some channels in it. Inside, if you take a cross section, it has some channels. So into this channel, the DNA molecule that needs to be transcribed goes inside. So in the channel, you have the sigma factor. Already you have seen sigma 4 that binds to the minus 35. Then you have the sigma 1.1 and so on. There are different sigma domains I told you. They go and associate with these minus 10 and minus 35. So the DNA molecule gets threaded across the polymerase. It is holding it. Now the structure also shows you the DNA is opened up. The process is called as melting. If everything is fine, if the polymerase, the sigma factor has gone and attached to the promoter correctly, and if everything is ready, the DNA is opened up. The process is called as melting. I'm not going deep into how it opens up and so on. It is again interesting molecular mechanism, because since it is little off the purview of our discussion, I'm not stopping there. The polymerase changes its shape itself so that the DNA molecule is wrenched open. It is called as melting. So once the DNA opens up, now transcription begins. So this process is called as initiation. The initiation is nothing but the the promoter needs to be identified. You understood what is the structure of the promoter. The polymerase goes and binds to it through a sigma factor. Once it is bound, the closed complex, when the DNA is not melted, the closed complex becomes an open complex. When the closed complex becomes an open complex, now elongation begins. So see here, in prokaryotes and the eukaryotes, the function is quite same. But the proteins that are involved is little different. It is more elaborate in the case of the eukaryotes than in prokaryotes. So to give you an idea about what is the complexity, see here. So these factors that are required for transcription is called as general transcription factors, or GTFs they are called. So the GTFs comes in different names: TBP, TF2A, TF2B, TF2E, TF2H, S, and so on, TAFs, and so on. I'm not dwelling deep into it. So these proteins are also required for initiation to begin. The initiation is little complex here in the case of eukaryotes. So listen very carefully. See here, just I'll walk you through the process. I'm not going to delve deep into it. The same process, but it is affected by different sets of general transcription factors. For example, see here, you have a Tata box. The Tata box belongs to the promoter. So the Tata box is initially identified by the transcription factor TF2D. It has a Tata binding protein TBP. It goes and identifies to it. Then another 2 protein goes and identifies the promoter. So it is not single identification as in case of prokaryotes. Different proteins, they get identified. Different proteins, they identify the promoter. Once the promoter is coated with correct protein, the polymerase is recruited. The word is called as recruitment by TF2F. It recruits, brings the polymerase and attaches to the promoter. Now you can see the promoter also has an interesting tail. It is called as a CTD domain. C-terminal domain it is. Don't need to worry about too much of it. That domain is important for stabilizing the binding. So every protein sits on the promoter, the CT domain goes and stabilizes it. Once it stabilizes it, you can see the other transcription factors they attach. So you can see the DNA is closed here. It is a closed complex. But when TF2H along with TF2E goes and binds to it, the structure of the polymerase, they change. The DNA opens up, open complex. Once open complex is done, the DNA starts moving. Sorry, the polymerase starts moving. Sorry, the polymerase starts moving. Elongation phase. So don't need to confuse yourself. This on the tail you have a series of phosphate. It is called a phosphorylation. In biology, certain proteins can be functionalized and dysfunctionalized by addition and removal of protein, addition and removal of phosphates. The enzyme that adds a phosphate is called as kinases, and the enzyme that removes the phosphate are called as phosphorylases. So an interplay of these two enzymes functionalizes and dysfunctionalizes something. So see here, when the polymerase has to move away from the promoter into the other regions, transcribing it, the tail should lose its affinity for the other proteins and move. So let me sum up what initiation is all about. Initiation should include a sequence upstream to the gene that helps the polymerase to identify that promoter. Once it has identified it, closed complex become open complex, then elongation. This is what initiation is. Next, listen very carefully. For example, if you take a RNA molecule in a eukaryotic system, an mRNA molecule in a eukaryote, you know that it is structurally different from the prokaryotic mRNA. In the 5', you have a methylated guanine. So the 5' always has a guanine. It is methylated, a methyl group is attached, CH3 is attached. On the 3' end, it has a series of adenine, poly-A tail it is called. Polyadenine tail it is called. So modification happens during transcription itself. How does it happen is the question. I told you that is where the tail of the polymerase. What is the tail called? CTD tail or C-terminal domain tail. So for example, see here, when the polymerase is moving, for example, the tail gets modified differently. In this case, 2P has been indicated here. When it is modified differently, it takes up a particular enzyme, capping enzyme. The capping enzyme is responsible for 5' cap. So as the RNA, you know that the first end that comes out is the 5' because the direction of synthesis is 5' to 3'. So once the initiation is over, when it goes into elongation, the tail gets modified little differently. The capping enzyme goes and binds to the tail, piggybacks on the enzyme. Then the outcoming RNA is modified by adding a methyl group to the guanine. Now, when it is running, probably you might find it difficult to understand. The gene in a eukaryotic system has two components to it. One is called as exon, the other is called as intron. Those genes are called as split genes. It is interrupted. You have a gene, it has coding sequences. The sequence that makes sense are called as exon. The coding that doesn't make sense, which is called as nonsense, it's called as an intron. So you may ask why there is intron at all. It's an interesting discussion. But introns are there. Surprisingly, 90% of the genes that we have, 90% of the DNA that we have are nonsense, junk. Only 1 to 10% codes for something. It's interesting. But whatever it is, the junk has to be removed and the information has to be connected together. So that has a series of enzymes which is called a splicing mechanism. What is splicing? Cutting something in budding or in making hybrids of roses, people cut one of the stem and add to the other stem. The process is called as splicing. Likewise, the ends of the introns are cut, the introns are removed, the exons are connected together. I am putting that in a simplistic fashion, but it's interesting, little complex mechanism. But remember, what is really happening? When it is moving, it is getting the 5' is getting capped. As it is moving further, the introns are getting removed. Then finally, when it reaches, you can see the phosphate or the phosphorylation pattern is slightly different. You have the polyadenylation factors gets incorporated into the tail, and it is responsible for adding a series of adenines to the tail. So once the transcription is over, all modification is over. That's how we need to understand. So see here, elongation we have understood. So see here, promoter. You need to identify the promoter. It's little simple in prokaryotes, little complex in eukaryotes. Closed complex, open complex, and elongation. Elongation, capping, splicing, polyadenylation. Then it has to stop somewhere. The process is called as termination. Termination is really interesting. There are two types of termination based on the requirement for a particular protein, which is called as a Rho protein. Rho protein, as it is indicated, 1, 2, 3, 4, 5, 6. It is made up of hexameric subunit. It's an hexameric protein. Rho protein it is called. So Rho protein is nothing but whenever the polymerase reaches a termination site, it is a particular sequence. The Rho protein goes and attaches to it. Once the Rho protein associates with the newly synthesized RNA molecule, it changes its conformation and pulls it, wrenches and pulls it out of the polymerase. Done. RNA is removed now. RNA goes into the cytoplasm. So this process is called as Rho-dependent process. Rho is a protein. But there is another mechanism which doesn't depend on Rho. It is called as Rho-independent protein. That's a beautiful mechanism. What I told you, the knack is in pulling the RNA out from the channel. I already got introduced you to the channels. Pulling out the RNA molecule, the newly synthesized RNA molecules from the channel is what termination is all about. The pulling, if it is aided by Rho, you call it as Rho dependent. If it is not aided by Rho, it is called as Rho independent. I'm not sure you people will be able to understand this, the Rho independent process. Listen very carefully. The DNA molecule is being transcribed now. Now the RNA is already synthesized. It has reached the termination point. Now there is a site at the termination point. I'll give you some description of that site. This is the life promoter. It has a particular sequence pattern. It usually shows a dyad symmetry. Dyad meaning the two sequences are complementary to each other. So it produces a hairpin. So see here, I'll show you this one. Hope you're seeing the cursor. So this sequence and this sequence are complementary. The blue colored sequence are complementary. So they can bend into a hairpin bend. The C pairing with G, the G pairing with C, G pairing with C, G pairing with C, and C pairing with G, and so on. It forms a loop. Now listen very carefully. I'm not sure I'll be able to convey it, but this is what beautifully happens. The polymerase is moving and it hits the termination site. What is the termination site? This is the site. Now it crosses that site. Once it crosses the site, that sequence comes into the RNA molecule because it is also transcribed. The sequence comes into the RNA molecule. Now I told you they are complementary. The RNA very quickly bends into a hairpin shaped structure. Don't you see this? It violently folds into a hairpin shaped structure. We people call it a secondary structure. The secondary structures fold so violently, it pulls the RNA out from the channel of RNA polymerase. Stopped. Transcription is stopped. So this is how termination happens. Row dependent and row independent. Now let me tell you, there are three phases in transcription: initiation, elongation, and termination. Initiation, you know that you have to have a start signal. Elongation, moving from closed complex to open complex. Then in the case of eukaryotic RNA, capping, splicing, and polyadenylation tail. Then it reaches a termination site. The termination is affected by the row protein in row dependent processes. In the case of row independent processes, it is carried out irrespective of the absence of row by the formation of secondary structures. Is that clear?
Sir, I think there is yes coming up in the chat box.
Okay, got it. Okay. Ma'am, how much time do I have?
No, it's like you can just continue, sir. Maybe another half an hour also. I think students will be here only.
Okay.
Okay. So, sir, maybe it's like you can cover the key concepts, and meanwhile, I think there are some questions.
Okay.
One question is from Khushi. Is it the RNA polymerase which moves ahead while reading the DNA strand, or is it the mRNA which moves while the reading?
No, it is the RNA. It is the polymerase that moves ahead.
Okay.
As the polymerase is moving in the active site, the polymerization happens. As the RNA molecule is, then I forgot to mention an important distinction between replication and transcription. Both of them, of course, you know that the product is nucleic acid. But in the case of replication, you know, both the strands, they separate. You know that. It is like opening a zip. But in this case, you can see as the DNA is opened up, from the rear, the DNA is closed. So it is the bubble that is moving. Only a small area that is continuously opened up is moving from promoter to that of the terminator. So it is local. It is not global. It is local. So the melting is always a 10, 15 nucleotide long. It starts moving like this. So that is one important distinction. Now, very quickly, there are many things that I can, I feel like talking to you people, but that's okay. Let's have a very rough description about, so that if at all, if all of us, if some of us are inspired, you know, we can embark on a learning process, a self-learning process, I would say. Now, listen very carefully. I wanted to talk to you also about translation, but on a later date. But I'll stop here after discussing about the way in which information is stored in the DNA molecule or in the nucleic acid. So listen very carefully. Central dogma. The information in the DNA is converted to the information in the RNA. You call that as transcription. Roughly, we understood what transcription is all about, how interesting it is. Then the next is what is called as translation. So translation actually converts the nucleic acid information into protein information. So it is interesting to understand how the information in the nucleic acid is converted to protein information, because the proteins are made up of amino acids and the DNA is made up of nucleotides. Then there are 20 types of amino acids. 20, 21, 22 is what we have, but those are modified amino acids, selenose, cysteine, and so on. but 20 amino acids, standard amino acids. The 20 standard amino acids has to be read from the information written in nucleotide. But you know that there are only 4 nucleotides: adenine, thymine, guanine, cytosine. But there are 20 amino acids. But what about 2 letters? Is the question. So 4 into 4, 16, still it doesn't cover 20. So probably a combination of 2 nucleotides is also not sufficient. So people, even before understanding how the information is stored, people understood that it should be a nucleotide of 3 that codes for, because 4 into 4 into 4, because you have 4 nucleotides, 4 into 4 into 4 is 64, which is higher than 20 amino acids. So if at all, if nucleotide has to code information for single amino acids, it has to have it has to be represented in sufficient number. So people understood that the code should be a sequence of 3 in order to code for 20 amino acids. But there is an issue. There are only 20 amino acids, but you have 64 codons. The 3-letter designations that code for an amino acid is called as codon. But 20 amino acids, you have 64 codons. But people later understood, by understanding the molecular mechanism, they understood that 3 of the codon do not code for anything. They do not code for amino acid. They later understood they are required for stopping coding. They are called as stop codons. You see a stop codon, 3 of them there: UAA, UAG, and UGA. Ochre, amber, and opal. Based on the semi-precious stones, they have named it as ochre, amber, and opal. So 64 minus 3, 61 amino acids. 61 codons, sorry. Codes for 20 amino acids. Clearly indicates that there are synonyms. Meaning, there are some slight variations of this codon coding for the same amino acids. So you have multiple codons coding for single amino acids. So this property, people call it as degeneracy. It is not normal. It is little abnormal. Hence it is called as, it seems to be degenerating from normality. Instead of having 20 amino acids, instead of having 20 codons, you have 61 codons. So you have multiple codons for single amino acid. So see here, UUU codes for phenylalanine. UUC also codes for phenylalanine. Both these codons are called as synonyms. You have UAA for leucine and UUG for leucine again. CUU leucine, CUC leucine, CUA leucine. So you have synonyms. So which codon codes for which amino acid is actually deciphered by, we can be proud in a way, but he was denied of the Nobel Prize and so on. Nierenberg, a German, and Khorana, an Indian. So they cracked the codons. It's really interesting to study how did they crack it. They made some synthetic RNA molecule with particular codons in it, then found out what sort of protein it codes. Interesting experiments they are. So they deciphered what codon codes for which amino acid. This is the codon chart. So all of you might know how in your smaller classes you would have studied. So this chart, it's a matrix. So this side indicates the first codon. This side indicates the second codon, and this indicates the third codon. So U, first codon, the U, the second codon, and you put the U here, third codon. So you have U, U, U. So that's how this matrix has been filled. So see here, I told you there are 61 codons, 20 amino acids. There are synonyms. And interestingly, these codon rules seems to be universal. Till now, it is universal, meaning this codon chart is followed by almost all the biological system. But I told you biology is also about exceptions. You cannot tame biology by certain rules. That's why you don't have laws in biology, You have only theories, hypotheses, dogmas, and so on, because we might not know when will we jump on an exception. So right now it is universal because it is found in all of the biological system. But I told you there is an exception. You have in a biological system the powerhouse of the cell, the mitochondria. In the mitochondria, the codons are different. They do not follow the chart, the standard chart. That's what I told you, it's an exception. But the standard codon chart is universal. Second, so what are the two things that I talked about this codon chart? It is universal. It is degenerate because you have synonyms. Degeneracy is explained by a theory called as wobble hypothesis. It is by a theory called as wobble hypothesis. The wobble hypothesis is also credited to Francis Crick. What does the wobble hypothesis say? That in most of the cases, the third codon seems to be wobbling. Wobble meaning when you spin a top, the top wobbles to its sides, or a gyroscope wobbles to its sides. It's called wobbling. So the third codon wobbles. The wobbling hypothesis also you can molecularly explain it. If people are interested, you go and study more on how the tRNA molecule accommodates this wobbling. But let me explain what is wobble hypothesis. Wobble hypothesis says that in most of the instances, the first new nucleotide, most of the instances, first two nucleotide seems to be coding for something, and the last, the third one, wobbles. So that wobbling doesn't have an influence on the type of amino acid it codes. I can give you an example. UUC, UUA, and UUG, or UUA and UUG, leucine and leucine. The first two nucleotides, they are the same, but the last one wobbles. Even if it is A, it is leucine. Even if it is G, it is leucine again. So the synonyms are built like that. So CAA, CAG, glycine, glycine. Even if A is converted to G, no issues. But there are many other combinations here. But wobble hypothesis explains why the codon is degenerate. Then the last one is codons are non-overlapping. What do I mean by that? So you have two codons, imagine. The codons are not overlapping, meaning one codon is red and after the next codon is red. Overlapping meaning the codons are overlapping. The codons are overlapping. Meaning, I'll tell you, where I can write, annotate. Okay. Imagine you have A, T, T, T. I shouldn't have put T. It should be U. It is RNA, is it not? U, G, G, G. Imagine this is one rough sequence. So the codon is read like this: A, X amino acid, U, Y amino acid, G, M amino acid. This is non-overlapping. First 3 codons are red, the next 3 codons are red, and so on. What is overlapping? This one is red, then these 3 are red, and then these 3 are red, and so on. So it is overlapping. So the codons are non-overlapping. But beware, there are exceptions. In certain viruses, it is overlapping. But generally speaking, they are not overlapping. So it is really interesting how the codon seems to be universal. So what does universality meaning? Right during the beginning of the evolution itself, the codon chart was set. The nucleotides have sort of come to a consensus that this should be the codon chart. That's why in all organisms, you have this codon chart. Everything that evolved from the first organism has this codon chart, not being replaced, not being changed, not being deviating. Are you getting my point? So shall we stop here? Lakshmi ma'am?
Yes, sir, I think so. So students, I
Okay.
would request if there are any queries, can you please type that in the chat box so that sir will be able to help you. And I hope the biology topics that you have been asking for is covered as of now. And if you have any queries, please do type it.
Sriram has typed in what is nucleoid. Nucleoid is basically a modified cytoplasm. It is not a proper nucleus. It doesn't have a membrane and so on. The cytoplasm is little concentrated there with specific molecules being concentrated, especially those associated with replication and so on. It is a slightly designated area. For example, you have a proper restaurant with tables laid and so on, with curtains and so on, or you can have a corridor with few tables placed there, and it doubles up as a restaurant. So nucleoid, in fact, is a rough compartmentalization for replication to happen.
So, sir, I think there are no further queries from the students. And dear children, please note that joining link to the refresher course remains the same on all the days. And
okay.
Yes, we'll be posting these videos in the YouTube, and then we'll be sharing the link towards the end of the session. So yes, I think we have covered all the queries and everything. So yes, I think it's time to wind up. Yeah. Thank you so much, sir.
Okay.
And
thank you so much, ma'am. Thank you so much.
And I hope it was helpful to the kids also, and we'll provide you with the recordings. So thank you all. Have a good evening.
Thank you, ma'am.
Yeah. Thank you.
Thank you. Thank you. Bye. Good night.